Well, it's 12:30. Time to start. We have 25 people. First, I'd like to remind you that you can get credit, extra credit in this course by participating during class. That means speak up. And for each class that you do that, you get 3 credits, which by the end of the semester accumulates to about 10% of your grade. So I do this to encourage you to talk, participate, ask questions, give your opinions, whatever's on your mind. There are 2 things I want to remind you of. One is you have a worksheet that's due Sunday night, and we'll discuss it next Monday. Some of you have already completed that worksheet, and you did very well. The other thing is that on Monday, there will be a quiz. Quiz is worth 10 points, and it takes 15 minutes. That will be the time limit on it. And you'll have all day Monday to do it. So from midnight to midnight, you can take your 15 minutes and do the quiz. And then it will be graded after that. Any questions about the quiz or the worksheet?
Can you hear me?
Yes. One thing about participating, you have to give me your name.
Oh, I'm Elizabeth Ortiz.
Elizabeth Ortiz. Okay.
I was wondering, at the end of the worksheet on the last 3 questions, it says that it's class notes, but we're not going to talk about it in class, or are we?
Yeah, we will today.
Okay. Just making sure.
Quick question. It's Alex.
Okay.
Alex, can you hear me?
Yeah.
For the quiz 1 for Monday, will you be telling us what like materials we need to study or know? So before we take it, like, will it tell us like, here's what you need to know?
I can tell you right now.
Yeah.
You need to look at the slides, the last slide set on Monday and today, and look at the worksheet, and that should cover it. So the quiz will
had from
cover.
Monday, right?
Pardon?
The worksheet we had from Monday, right?
Yeah, the worksheets actually due it Sunday night.
Okay.
But we'll discuss it on Monday.
Okay.
And if you have done the worksheet, that will help you with a couple of the questions on the quiz.
Okay.
I have a quick question.
Yeah, your name?
Oh, Yusuf Faraj. My bad. What else is due on Sunday? Because for some reason, nothing's like really popping up on my calendar. And everything's kind of a link in the module, so I don't really like know what's actually due on Sunday. I mean,
You should have an assignment. If you look in assignments, you should see the worksheet. I haven't really updated the calendar, and thank you for reminding me. I'll try to do that.
I don't know. I was just wondering. Thank you.
Yeah, if you can't find it, let me know.
Okay.
My name is Wassan Rafo, and I have a question about the worksheet. My question is about the terms, the definition. It should be like my own word or how it is like in the book.
You can do either. I prefer that you follow the book, but if you want to put things down in your own words, I give you credit unless you're wrong.
Mm-hmm.
So for any correct information, whether it's identical to what the book says or not, I give you full credit. but only if you put down something that violates the facts, do I deduct points.
So it could be like almost exactly how is like defined it in the book.
Yeah,
Okay. Another question is, when I looked at the syllabus, I saw that I couldn't find the syllabus there. So I don't know. I saw that just the assignments that we have to do, like the quiz, and the worksheet. I didn't see the syllabus page.
okay, you can always find things under files. But the syllabus, I think, and someone can correct me if I'm wrong, but I think you'll find the syllabus in announcements. But it reminds me, I should put the syllabus in module 1. So if you click on the home page, and I'll do that.
Oh, okay.
Professor?
Yeah, your name?
Hi, Fakriya. Abdul Sattar.
Okay.
So I was starting yesterday to do the worksheet, and I saw there is so say like on notes, but when I check the video, there is something missing. So what we should do, like if there's something missing, it doesn't explain on the notes, so where we have to look it up.
So there's something missing in the video recording?
Yeah, I mean, like, it's not explain everything for the notes. So what if there's something missing? So I don't know, like, we have to look in the book or.
Well, you might, yeah, several of them gives you page numbers to look at in the book. And for some of it we'll talk about today.
Okay.
So when you look at the recording of this class period, you should find any answers that you don't find in the book or from last time, you should find today.
Oh, okay. Thank you.
All right. I will go on and share my screen with you.
Professor,
Yes.
this is S. Steve Stinson. I forgot what link it was for going to the book.
The link, you'll find the link for the book in the syllabus. So just find the syllabus and the link is right on the first page.
I think we can also download the book if it in our computer. It's easy.
Yeah, it's easy to download the book. The link is right there in the syllabus. And if you click on the link for downloading, it should come right into your computer.
Unfortunately, on mine, I'm not getting the link. As a matter of fact, in the syllabus, I'm only getting, when I go to syllabus, it says, gives you the date, Sunday, August 22nd, and then it says worksheet introductory to biology, and then quiz one living thing intro, and then it says, do you want to study or learn?
Okay, you're not getting the syllabus. All right, I'll make sure that the syllabus is posted in module 1, so that if you click, so after the session today, if you click on home, it will default to module 1, and you'll see syllabus there.
Okay.
Syllabus, it's on announcement.
Yeah, you should be able to find it under announcements, but if you don't, I'll make sure that it's in more than one place.
Yeah, I didn't see it under announcements, but I did find the link for it, so I'll put it in chat for anyone who needs it. This is Anthony
Anthony,
MLO.
okay. All right, good. Thank you for doing that.
I'm assuming you already took attendance, Professor?
I don't take attendance. I record participation.
Right.
So that's why I ask you for your name anytime you speak up. I've already got your name for today. So whenever you speak, give me your name and I'll record it that you have participated, and then you will get credit for that.
Okay.
Now you should see on your screen the methods of scientific inquiry. Anybody not see that? Say so. Otherwise, I'll assume you can see it.
Yeah,
Yes.
I can see it.
Okay,
Yeah,
see
I
it.
can see.
good.
I can see
Okay,
it.
thanks. And this slide set is all we're going to do today. So how do scientists investigate the natural world? What methods do they use? There are many. Requires logic, analytical skill, and especially perseverance. You have to stick to it if you want to investigate natural phenomena. Biological systems are very complex. You've probably somewhere in your past been exposed to the so-called scientific method. And it's usually presented something like this linear arrangement where you first make an observation, then based on that observation, you make a hypothesis. And then after stating your hypothesis clearly, you set up experiments to see if your hypothesis is true. Could you mute your mic, whoever's got background? Now, if the experiments support the hypothesis, you do more experiments, and if you can gain enough evidence, you make a theory. However, if the experiments don't support the hypothesis, then there's something wrong with the hypothesis, and you go back and make a new hypothesis. Similarly, if the theory under further investigation turns out to be violated, then the theory is wrong, and you go back and make a new hypothesis. So this is nice and neat and straightforward, but science almost never works this way. And I'll come to that in a minute, but first, there's two different types of investigation when it comes to experiments. So-called curiosity-driven investigation and applied research. Applied research is the practical application of scientific methods to specific everyday problems. You have a problem, you use scientific methods to address that problem. A curiosity-driven investigation doesn't really have a goal. It's, I wonder what would happen if I mixed red paint with green paint. You don't have anything in mind that you're going to do with this mixture of paint. You just wonder what would happen if you did it. What if I put vitamin pills on my plants? Or poured sand in my gearbox? Or what if I fused hydrogen atoms together? Or whatever you can think of, whatever you can imagine would be classified as curiosity-driven investigation. So how does science really work? these 4 boxes illustrate components of scientific investigation. You're familiar with these: making observations, asking questions, hypotheses, thinking critically, communicating the information that you get, conducting investigations. Now, what you see is that these 4 boxes are all connected to each other by double-headed arrows. They point both ways. That means that any one of these could be a starting point, and your investigation may proceed in any of these different directions. And this, some of you asked about question number 6 on the worksheet. This is it. This is how you answer question number 6.
Quick question, Professor.
Your name?
Alex,
Okay. Yeah.
on the communicating like information, is that like, would you say like, are scientists just communicating with other scientists? Is that what you mean?
Yeah, good question. Communication takes many forms. And I have a slide later which outlines some of those forms. But yes, talking to other scientists is probably the primary way of communicating. You meet somebody in the hallway, or you have a phone call and you communicate information that way, or you can speak at a meeting. You may have a seminar. You may go to a conference. There are a lot of different ways of communicating scientific information. So we'll talk about that a little bit later. Is there a single scientific method? Here are the 4 components again, but I want to emphasize the one in color: testing reproducibility. So you can get a particular result, and it may be exactly what you were looking for. But if you do the experiment again and you don't get that same result, then it's not reproducible. So the first thing to keep in mind is you have to be able to repeat experiments in your own facility. And then secondly, and equally important, other scientists in other facilities, other locations, have to be able to take the information that you communicated and repeat the procedure exactly as you did and get the same result that you did. And if that happens, then you're on your way to a theory, because it's reproducible. You do the same thing over and over again, you always get the same result. If you try to repeat and don't get the same result, then something is wrong.
Professor, I have a question.
El Arredondo.
Yeah, Angel.
I got you.
How many times do you have to repeat it so it's credible, like amongst everyone? Is there like a bottom, like baseline, like I don't know, like at least?
There's no fixed number. If you can repeat it once, that's good news. If you can repeat it 10 times and you get exactly the same thing every time, that's pretty convincing. But if somebody in another lab can do it repeatedly, then that's very convincing. So yeah, there's no particular number of repeats that you have to do. You really have to convince yourself. And once you've done it enough that you're convinced, then you publish it or communicate it in some other way and see if other people can get your result. Okay, here is a really simple example, everyday example of observation and hypothesis testing. You flip the switch, nothing happens. The light is out. So you have a number of hypotheses. Could be that the bulb's burned out, could be the power to your house is out, or maybe the breaker has tripped. So how do you test these hypotheses? Well, you can replace the bulb. If it now lights, then the result is yes, you've solved the problem. If that doesn't help, then check other electrical appliances. If none of them work, then very likely the power is out. But if other appliances don't work either, then you get in touch with the power company and say something's wrong. Similarly with the breaker, if you can flip the breaker and everything works, you've solved the problem. So this is just a really simplified but real example of how we do hypothesis testing, often in our daily lives. Well, there are other methods besides the so-called scientific method. And here I contrast two of them, how scientists work as opposed to engineers. So scientists, starting at the top, ask a question. Engineers define a problem. In both cases, they do background research. Scientists construct a hypothesis. Engineers specify requirements. Scientists test an experiment, whereas engineers brainstorm with other engineers. They evaluate data, historical data, and they choose a solution. Then there is is the procedure working? Test solutions, but at the end, in either case, the results are communicated. So I show you this not because it's in any way profound, but simply to make the point that different types of professional people pursue investigations in different ways. If you wanted to look it up, lawyers have yet another way of doing investigations, and so on. Okay, making observations and asking questions. Questions arise as a result of observations. And questions can be answered by either making observations, which is called discovery-based science, or via experiments, which is known as hypothesis testing. So these two are the two basic ways of gathering information. So a hypothesis is a tentative explanation or an answer to a question. And you can address that either experimentally or observationally. So if you do an experiment, what is that? What is a controlled experiment? A control is a constant. Throughout the experiment, it does not change. So for example, in agriculture, if you want to know the effect of fertilizer, what do? What is this?
Just grass?
Some kind of grass, yes. You see any difference in the two sides of the slide?
Yes.
One side is more
Yes.
full, and the right side seems like there's emptier patches of grass.
Right. Well, the right side, the grass, these are actually wheat plants. The wheat is doing better on the left than it is on the right. And why do you think that might be?
Fertilizer.
Angel, I think.
fertilized.
Okay, Angel,
Yeah, I think one side is like watered more than the other.
could be that.
I think it could be the nutrients maybe in the soil breaking up.
Maybe that too.
Perhaps one is more controlled than the other.
Uh-huh.
It could
Um,
also be like the sun is facing one direction more than the other two.
Yeah, possible, but since this appears to be
Could be
just,
one is more fertilized and the other is not.
that is in fact the case here. Some of your other suggestions are possibilities, but this picture is taken from an experiment in which fertilizer was being tested. So the ones on the right did not get fertilized, the ones on the left did. So go back to our example, effective fertilizer. What is the control in this experiment?
The amount of fertilizer. This is Steve.
Steve Simpson, okay. All right. The amount of fertilizer? No, that would be a variable.
Oh,
I want
the
to make
control is the wheat.
Could it also be the strength of the fertilizer?
No, it's, I just realized. I got it wrong, I know now.
The
Okay,
control is the weeds that never have fertilizer.
right, that's the constant. So the control, if we go back to here, control is a constant throughout the experiment. It does not change. So if you're testing fertilizer, one thing you don't change is a treatment that has no fertilizer. We'll talk more about that later. Another example would be in medicine, if you want to know the effect of a drug. What would be the control if you're testing drugs?
This is Steve
The amount
again.
of the amount of it.
The group that is not receiving the drug is the control group.
Right. You have to have a group, as you said, a group that doesn't get the drug. And then you look at people who did get the drug and had the same condition, and you find out if it had any effect.
I have a question, Professor.
Rodriguez, Samuel, is this Samuel?
Yeah.
Okay.
Can it also be a part of the control that all the wheat is like planted at the same time as well?
Yes. Yeah, that would be critical. You have to, that's part of the constant. That all the wheat is treated exactly the same, except for the fertilizer in this case.
This is Faith Davila. I had a question.
Uh-huh.
So in general, though, the wheat would be what? Since it's not like it just the constant in general, like both sides?
Okay, ask your question again.
Like just in general, because both of them are wheat, is what I'm saying. Like, what is the plant? Would it be like the constant?
They're both wheat, right? So all the same seeds were planted in this entire plot, but then a line was drawn down the middle, and the plot or the plants on the right were not fertilized. The ones on the left were. So the constant here, the control, is the plants that weren't fertilized. And then, as somebody said, for those that were fertilized, you could have any number of variables. So for example, you could try different types of fertilizer, or you could take a single type of fertilizer and vary the amounts. And I'll talk about that in a little bit. It'll become clearer as we go. Back here. Okay, observational science. Investigations may or may not be testing a hypothesis. So for example, if you're a geologist, you might observe rock formations. You don't do any experiments on them. You could, but not necessarily. And if you don't, if you just observe rock formations and compare localities, that would be one way to do observational science. Another example in the social sciences, political science, it's very difficult to do experiments on humans. So political scientists rely largely on historical precedent. They observed what has happened in the past. And many questions are addressed answering using both of these methods. All right, an experiment is a controlled test. One variable should be changed at a time. And there are two types of variable. An independent variable, which is changed by you, the experimenter, and a dependent variable, which is the response to the independent variable. So let's go back to our example of fertilizer. We think back of on the picture here, the independent variable is the fertilizer. So the plants on the left are the independent variable. The ones on the right are dependent. So the application of the fertilizer X in this experiment is the independent variable. The response of the plants depends on how you apply the fertilizer. And at the bottom here, what you see.
Professor, I have a quick question. It's Alex Alos, by the way. Why only one variable changed at a time? Why not just maybe two sometimes?
Well, you can't change two variables at a time, but you can have different variables. So you can have one control, for example, no fertilizer, and then for the experimental, you might have fertilizer A or fertilizer B or fertilizer C, in which case you're now comparing the effect of fertilizer type with no fertilizer at all. Or you might vary the amount of fertilizer. So if we go to the bottom of this graph here, we have an x-axis and a y-axis.
Professor, this is not taking into account the amount of water. If that's just another control variable, let's say.
Yeah, they would all get the same amount of water.
Okay.
Having trouble with my slides here. Okay. All right. So when you plot data on an XY graph, the y-axis is vertical, the x-axis is horizontal. The dependent variable, so I can get rid of this. The dependent variable is always on the y-axis, and the independent variable is always on the x-axis. So in the case of the fertilizer example, the amount of fertilizer goes from low to high. And as the amount of fertilizer varies, plant growth varies from low to high. So the more fertilizer you put on, the more the plants grow. So remember that. This will be on the quiz on Monday. Plotting dependent and independent variables on an XY graph.
Can you repeat that again? This is Steve again. Can you repeat that statement one more time?
Okay, the thing to remember is that the independent variable is always on the x-axis, and the dependent variable is always on the y-axis. And in this case, the amount of fertilizer on the x-axis, that's the independent variable. That's what you can control. You control the amount of fertilizer. What you don't control is how the plants respond to it. So the plant response is on the, that's the dependent variable, depends on what you do, and that's plotted on the y-axis. Oh, here, okay, I got rid of that. So now you can see the x-axis and the independent variable at the bottom.
I have a question. This is Alicia Carroll.
Alicia Carroll, okay.
Is there ever an instance when the independent variable switches with the dependent variable, or is it always the same throughout the scientific method?
Do they ever switch? No.
Do they ever switch? Okay, thank you.
Okay, here's some examples. Observational, experimental. A scientist goes to Alaska 3 years in a row, counts the number of polar bears found in a particular 5 square mile region. Is that observational or experimental?
Observational.
Observational.
Say your name.
Priscilla.
Uh-huh. And you said it's which?
That it was observational.
Observational, okay. Anybody disagree with that?
No.
Okay.
No, no, I
Um,
don't.
All right, that's correct. This is observational. All this scientist is doing is counting. Look at the next one. A scientist determines the DNA sequence of the gene responsible for cystic fibrosis. Is that observational or experimental?
I want to say experimental.
Okay, what's your name?
Alex.
Alex, okay. Alex says experimental. Anybody agree with him?
I agree with you, Alex.
Okay, got an agreement.
Gabriela Galvez says it's observational.
Gabriella, okay, Galvez, all right. You think it's observational? All right, anybody agree with Gabriella?
My name is Emmanuel, but you can call me Manny. I also agree with Gabriela.
Okay, so we have 2 votes for observational and 2 votes for experimental.
Make it a 3,
observation.
Professor. I agree with Gabriela.
Okay, there's 3 votes.
All right. I also believe that Gabriella is right. I think it's
Okay.
observational.
Okay,
Make that
It
5,
is.
Professor.
yes,
It's observational because it isn't a controlled test. Like there's no variables in it, correct?
yeah, that's correct. This is observational because what the scientist has done is observe the DNA sequence, the A's and T's and G's and C's in the sequence of the gene. There's no experiment done here. So if the scientist wanted to make this experimental, what might he or she do?
Professor, he should change, what's it called? A variable? Yeah.
Okay, change what? What would be the variable that he would change?
Professor is Manny. I'd say maybe having some sort of medicine for the disease.
Okay, that would be,
Professor, do you
yeah.
change a sequence in the DNA?
Okay.
DNA could be altered.
Now we're on the right track. Yeah. So if he gets a DNA sequence, and supposing that is from a normal person or animal, now the scientist could change that DNA sequence in the organism and ask, does that change in sequence make any difference? And we'll talk much more about this sort of thing later when we talk about genetics. But yes, you are right that in order to make this experimental, you have to change something and make a comparison. Compare one DNA sequence with a different DNA sequence. Okay, a scientist determines the rate of growth of plants given different doses of a new fertilizer. We've already talked about this, so this would be what?
Experimental. This is Lana
I
Burns.
agree. I would say it's experimental as well.
Yeah, and you're right, it is experimental. All right, scientists take a submarine to an area of the ocean floor that looks for different types of organisms. What is that?
Observational.
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 Right. It's observational, you all agree. There's no experiment involved. Scientists test the growth rate of a newly discovered bacterium on different types of food source.
Experimental.
Experimental.
Okay. So what would be the control in this experiment?
The type of
The
food source?
bacterium.
The food sources.
The
bacterium.
Food sources are being compared. Food sources are being compared, so those are variables.
Yeah, the common denominator is the bacterium.
Right.
The
Same bacterium is
growth
used throughout.
rate.
Yeah, this is kind of a trick question because you're comparing different types of food sources. You have to pick one as your control. You have to say, okay, here's my basic food source. I'm going to compare all these other sources to that. So in this case, it's up to the scientists to decide which food source is going to be the control.
Professor Yoder.
Yeah.
So I had a question. So you said you have to pick like a food source. Is there any chance that no food source will also be the control, or would you have to pick a food source?
Yeah, you're right. You could say no food source. And what would that be? If you were doing this in an agar medium, you could have just agar, but agar itself is an organic material and it's a food source. So it would be really hard if you did it in distilled water, nothing would grow. So to make this experiment meaningful, you have to pick a food source on which the bacteria could grow to some extent and then compare other food sources with it.
Professor,
Yeah.
does that mean that this one is observational because they're just doing something and see what happens?
What does anybody else think? Could this be construed as just observational?
It's probably a combination of both.
Yeah. Let's not belabor it. You are making observations, but you're also making comparisons. So when you make comparisons, you're comparing food sources. And so that is an experiment. And as I said, the experimental are the different food sources, but you have to have something to compare them with. And that's why I say at the outset, before you even start, you have to make a choice. You say, this is my standard food source. I'm going to compare all the others with the standard. Okay, we'll go on from there. Proposing a hypothesis. Hypothesis is a possible answer or a tentative explanation for your observation. It's different from a theory. A theory is a broad general set of principles. Hypotheses reflect past experience. You should propose multiple hypotheses if you can, because only one of them can be right. Now, here's something that will come up on your worksheet. What do hypotheses and theories have in common? They must, in both cases, they must be testable. and they must be falsifiable. That means it must be possible to disprove them if they're not true. So in biology, any proposition can be proven false. So you can prove something false, you cannot prove it to be true. So hypotheses can be eliminated, but they can never be confirmed with absolute certainty. There's no truth, no absolute truth. So let's talk for a bit about science and pseudoscience. Science changes with new evidence, whereas pseudoscience has fixed ideas that don't change. Science considers all the data. Pseudosciences focuses only on a portion of the data that support the conclusion that's desired. Science starts with an observation and ends with a conclusion. In pseudoscience, it's the opposite. It starts with a conclusion and then looks for evidence to support it. Science, you need to have repeatable results. Pseudoscience, non-repeatable. Science invites criticism. You want people with different approaches, different technologies to investigate your conclusion. Pseudoscience resists criticism. Science is driven by a desire to understand. Pseudoscience is driven by cultural goals or commercial or even ideological goals. Somebody has their mic on. We're getting some feedback here. Could you mute your mic, please? Now, I'd like you to think about this science versus pseudoscience in the context of what we see all around us every day. So I'll just give you an example, the first one here. Science changes with new evidence, as opposed to pseudoscience, which has fixed ideas. Now, we're in the middle of a pandemic, coronavirus. The CDC, the Centers for Disease Control, is a scientific organization that gathers data and evaluates it and puts out recommendation. Now, the CDC has come under a lot of criticism because it keeps changing its recommendations. Well, why does it change its recommendation? It's because the evidence changes. So as more evidence is accumulated, the CDC evaluates that and changes their recommendations. So even though it's confusing, especially to the general public, it's necessary to follow the data. You change as the evidence changes. And we'll talk more about coronavirus, for example, later in the course, but I just wanted to use that as a comparison for in our current culture. Some people are resistant to changes as evidence changes, and scientists, of course, are not. Now, how is a hypothesis different from a theory? Well, here's a table with criteria on the left, and then how a hypothesis varies from a theory in the other two columns. We've gone over much of this already. Hypothesis is a suggestion. A theory is well substantiated based on data, based on evidence and proven hypotheses. As I said before, both hypotheses and theories are testable and both are falsifiable. A hypothesis is not well substantiated, whereas a theory is well substantiated by a lot of evidence. Limited data for a hypothesis, a lot of data with a theory. Hypothesis is specific, usually based on one observation or a very limited number of observations, whereas a theory is the establishment based on multiple tests and experiments. And the purpose of a hypothesis is to present an uncertain possibility, whereas a theory explains a large set of observations. And some of this information will be relevant to both the worksheet that you're doing and the quiz on Monday. So you can go back over this and think about it by looking at either the slide set, which is in Canvas, or the recording of this class. Another issue that comes up from time to time is, how is a hypothesis different from assumption? Or are they the same thing? Well, in fact, they're not. A hypothesis is not accepted as true. It's proposed as one possibility, and it may or may not be falsified by testing. An assumption, on the other hand, is taken for granted. You believe at the outset that your assumption is true. It doesn't need testing because it's accepted as true. So an assumption is really not part of scientific thinking. Uses of the term theory. Theory as it's used in everyday conversation, theory as it's used in science. So in day-to-day speech, theory often means a guess or an unsubstantiated idea. For example, I have a theory that gremlins are hiding my car keys. In day-to-day speech, the word hypothesis is rarely used. In science, theory refers to a well-substantiated or even a proven hypothesis. Whereas in science, a hypothesis is simply a suggested explanation. It's commonly used at the outset of an investigation. So when you read the news or watch TV and you hear people talking about a theory, they probably really mean a hypothesis. Thinking critically. A major part of the scientific process. Identification and articulation of the problem. You have to define your problem clearly in order to investigate it properly. You have to gather all the relevant information, and you will do this in your cystic fibrosis project, where you will ask a specific question about your mutant, or a set of questions. You'll gather all the evidence that you can about your particular mutant. Organization and analysis of the information. So here I want to emphasize the importance of graphing your data. Graphing your data is extremely important. And it can make it much easier to understand. And to make that point, I show you these two data sets at the bottom. So look at the first one, the one on the left, and tell me, if you can, what are the differences between column 1, column 2, and column 3?
They all got different numbers. But it seems that column 1 has the highest number at the last.
Hey, Mr. Yoder.
Yes.
My name is Amor. For the first row, you could see it's kind of like it ranges from a high number and it ends at a high number. On row 2, it starts off at a low number and it rises, so like it increases over time. And then for the last one, it decreases from a big number to a lower number. So like the sequence of each rows are like different from each other. Or that's what I see.
Yeah, you can say that, but would you be confident in drawing a conclusion about column 1 with respect to column 2 and column 3?
Not necessary. This is as Steve.
I feel like there's
Stinson
not
again.
enough information.
Yeah, that's the point. The information is all there. It's just that you can't make sense of it the way it's presented. These raw numbers, they're in all 3 columns, they're just a jumble of numbers from, in some cases, very low to high. However, now look at the illustration on the right. What are the differences among the 3 animal species? Bears, dolphins, and whales. What are the differences?
A bear's on land
Bears, what?
or on land.
What we're talking about are numbers, numbers of animals.
They
So
dabble at, I think they are just fluctuating at different times. Like sometimes they almost like the bears and the whales are like almost the same, but like they mainly fluctuate.
okay, over time, what happens?
The dolphins decrease in wildlife population while bears, increase. And then I meant dolphins decrease. I don't know if I said that.
Yeah, you said it.
Dolphins decrease, bears increase, and then whales are like, they're stuck there in the
They're in
middle.
the middle, right?
Yeah, they're kind of in between.
Yeah, and you can say that with certainty, right? You're looking at the data plotted as a graph. And so within a few seconds of looking at that, you know the answer. You know that over time, between 2017 and 2022, which we're not there yet, bears increase, dolphins decrease, and whales don't change. So if you had taken the numbers, just the numbers, and plotted them in a table, you'd end up with the same kind of uncertainty that we looked at in this table on the left. The set of numbers is hard to interpret. If you make a graph, it's usually easy to interpret. And I show you these 2 illustrations just to make that point, that when you are doing your own investigations, graph the data. It doesn't have to be a line graph, it could be a bar graph, a pie graph, but a graph helps you to visualize the result much better than a set of numbers. Okay, someone asked earlier about how do you communicate. There are different ways to communicate. Talking to people is the primary way that scientists communicate. But once a scientist is fairly sure that his or her conclusions are correct, they want to publish. So there are a lot of different scientific journals, a lot of different books, like Science, Nature, the Public Library of Science. And when you will be investigating your mutants, you may end up looking at some of the articles that are published. Scientific meetings, discussions with other scientists. So yes, the point is that scientists have a lot of different ways of communicating their results. And now, thought problems. These are for you to think about on your own time. You can read through some of these. Think about fire. In what ways is it like a living thing? And in what ways is it clearly not a living thing? The second one is about starting your car or not starting it in the morning. think about the steps you would take to figure out what's wrong. The third one, a medical researcher wants to determine the effect of new drug in the treatment of disease. What would be a reasonable control group for this experiment? So you have a disease, diabetes, you have a drug, you want to know if that's effective against diabetes. What kinds of groups of people would you assemble to investigate this?
People with diabetes.
Right, that would be one group. And what would you compare that with?
Would it be with the old
People
kind of
without-
medication?
Okay, so you have people with diabetes, and what would you have, one group or more than one group?
Depending on how many
Groups.
drugs you're trying to test.
Okay, there's only one drug here.
2 groups, control group, which you would not administer the drug to, and the experimental group, or which you would administer the drug to.
Right, so you've got 2 groups of people, both groups have diabetes, and you give the drug to one of them. What other, so you have a treatment and a control there, what other control would you want to have?
Wouldn't you want to have the people that already are taking the old drug?
There's no old drug, there's only one drug
Oh,
in this experiment.
The amount,
The amount of the drug, yeah, that would be a variable. That you,
The age,
the age of the people, the
The
age
type
of the
of diabetes,
excuse me?
the type of diabetes, would that be one of them?
You'd have a standard type, it'd either be type 1 or type 2 diabetes. But
probably gender too, Professor?
yeah, but the assumption here, and it would be an assumption, is that things like age, gender, and so on would be normalized in the group, among the diabetes patients. So assuming that they are normalized, and so you've got 2 groups of people with diabetes, one of them is a control, the other one is a treatment. What would you like to compare those numbers with? How about healthy people?
The numbers before taking the medication?
Yeah, that's essentially the same thing. People who don't have
people
diabetes.
out.
You have healthy people and you divide them into 2 groups. And how do you treat them?
How do you treat the healthy people?
Would you just give them a placebo?
Yeah, that's a good control. So among the healthy people, what about the drug? Healthy people and the drug. Well, you want to know, does this drug have any effect on the patients, on people that don't have diabetes? In other words, are there side effects? And so if you have 2 groups of healthy people, one that treated with the drug, one treated with a placebo, is there any difference? So we're really talking here about 2 groups with diabetes and without, healthy. And for each of those, you divide them into 2 groups, one's treated with the drug and one isn't. I emphasize this one, or discuss this one, to make you aware that when you think about controls, there are usually, in most experiments, many controls, many things that you're comparing your treatment to. And in this case, there's only one treatment, and that is treatment of diabetic patients with the new drug. And everything else is a control. All right, going to the next one, difference between a guess hypothesis and a theory. We've talked about that. The next one is interesting. Consider the following proposed experiment. Explain why its structure is an example of pseudoscience. We want to study how global warming affects the growth of prairie grasses in the North American Midwest. What's wrong with that?
Is it because they're starting with an assumption, like they're already looking for a conclusion?
Yeah, you're on the right track. Just the way it's stated, it's stated as though they know that global warming has an effect. So it's stating a conclusion before they even do an experiment. So you're right. And then the last one is something for you to think about. We won't go through it here. Okay, is there anything that you'd like to go over, like to review again in this data set?
No, I think that was pretty much it. Thank
we're
you.
good. Thank
Okay.
you.
Well, it's recorded, and I will send you the link for the recording. And if there's nothing else, time is almost up. On Monday's session, it will only be 60 minutes because you'll have to take 15 minutes of your own time to do the quiz.
I did have one quick question, if you don't mind.
Yeah.
Faith, by the way. With the worksheet assignment, do you care, like, if I print it out and then I obviously write on it, do you want it like scanned, or can I take a picture of it and submit it, or what do you prefer?
I prefer that you upload it by one of the methods that's given in Canvas. I think it's either as a Word document or a PDF or text. I prefer that.
Okay, so you don't really want like your actual handwriting on it. You would rather it just be like typed out.
That's my preference.
Okay, that's all I was asking. Thank you.
Okay. But I should say that if you do have some reason why you'd like to write it out and send me a handwritten response, I will grade that just like the others.
Okay, thank you.
All right, anything else?
Nope.
Okay.
No questions. Thank you.
All right.
No, thank you,
all.
Professor.
All right, have a good
Thank
day.
you.
Thank you.
Okay, bye. See you on Monday.
Thank you. Bye,
See you
everyone.
Monday.
Thank you.
Nice weekend,
Have
Professor.
a good one.
Stay
Stay
safe.
safe.
Okay.
Don't do
Take
nothing
care.
I'll undo.